In our next IB video, we're going to look at membranes, and I've split this content into two different sections. So this is IB 2.4, and I've split this into two different parts. The first part will specifically look at the plasma membrane and the different components of it. In the second video, I'll talk about osmosis and diffusion, passive and active transport. It was just a lot of content to cover in one video, so I decided to split them for that purpose. So the plasma membrane is a very important part of the cell overall, and it's what allows or what controls what goes in and out of the cell, and it's actually a very complex organelle and part to the cell because it has a lot of different components to it. A term that you've probably heard before and that we use to describe the plasma membrane is the fluid mosaic model. And the fluid portion of that term means that the membrane is movable. It's bendable, it's flexible, it's kind of like a liquid in that it can move and bend and kind of change its shape. Mosaic primarily means because we have so many different portions of it. If you think of a mosaic in art, it's made up of lots of different pieces that put together make an overall image. And so in our plasma membrane, it's made up and it contains lots of different components, proteins and carbohydrates and phospholipids and cholesterol. There's lots of different portions to the plasma membrane, so that's what the mosaic portion means. And then model, we use models to help represent or to get an idea of what this actually looks like in real life. And so the plasma membrane is selectively permeable, and that means it is able to determine what goes in or out of the cell. Selectively meaning it's kind of choosy, permeable meaning controls what enters and leaves the cell. And so let's look at some of the different components or pieces that make up the plasma membrane. The first and the primary component is a phospholipid bilayer. And this is composed of two layers of phospholipids, which is phosphates and fats. The lipid is fat, and then phospho is the phosphate. And what this looks like is it has, I guess, two different portions. It has a nonpolar hydrophobic fatty acid tail. So this is the fat portion right here in the pink color. And this is hydrophobic, meaning that it doesn't like water. On the external portion of that, so the second portion of that, is our polar hydrophilic head. And this is where the phosphate resides. And this portion actually likes water. And so what we see is that it has a hydrophobic or afraid of water tail and a hydrophilic likes water head. And so this arrangement within the plasma membrane allows these hydrophobic fatty acid tails to be protected from water or an aqueous environment by the polar hydrophilic heads. And so if you can imagine that this is just a small snapshot here of the overall plasma membrane, if you imagine that this is a spherical, or it forms a sphere, a circle, all the way around the cell, this all the way around the cell, if I were to draw a circle here, this all the way around the cell, all of these fatty acid tails are going to be protected by these hydrophilic polar heads. Let me go back to our plasma membrane image here. Here's our phospholipid. So this is an individual phospholipid. The tail here is the fatty acid tail. Here's the hydrophilic head. And so if this were to extend all the way through, you can see all these phospholipids. In each case, the fatty acid tail is being protected by these hydrophilic heads. Okay? And so that goes all the way around the cell, and even though that it's bendable or flexible, or maybe there's some other components within the plasma membrane, these fatty acid tails are protected by the hydrophilic heads. Also, we will see cholesterol within the plasma membrane, and this is usually found in between, so it would be found in between phospholipids. And really what it does is it helps to reduce the membrane fluidity, movement in animal cells. It's found in animal cells, and sometimes it's important or necessary for the membrane to not be as fluid, not be able to move quite as much, and cholesterol helps to maintain that. So here's a phospholipid. Here's one phospholipid. Here's another phospholipid. And these little portions right here are cholesterol, and that forms in between the individual phospholipids and helps to add some structure and rigidity to the plasma membrane in animal cells. We also see glycoproteins in the plasma membrane, and obviously there's going to be a protein component to this. The glyco portion refers to a short carbohydrate chain that's covalently bonded to the proteins. And these often provide the cells a way to recognize other cells by binding to other molecules. So it kind of acts as like a communication network in the plasma membrane, and it allows cells to communicate with one another. Another portion is called an integral protein, and these are proteins that extend at least into the hydrophobic region of the plasma membrane, and they may also actually span across the membrane internally or externally. And let's look at what these look like. So going back to our glycoproteins, here's the protein component, here's the glyconutrient, this is the carbohydrate component, usually less than 15 sugars, and so the glyco portion extends outside of or externally to the cell. And you can see here are some examples. It allows this glyco portion is able to bond or connect with other types of molecules, toxin, virus, bacterium, other cells, and it allows them to communicate with one another. Obviously, a virus may not necessarily be good communication, but it does allow that. And then there's that protein component here. Integral proteins, like these here, they expand into the hydrophobic region, so in between the fatty acid tails, and they may actually expand across the membrane internally, so within the cell, or externally. There's also something called a peripheral protein, and these are proteins that are not embedded within the membrane, but they're attached to either external surfaces outside of the cell or within the cell, and attached to the surface of the membrane. Here's another image that helps to kind of clarify this a little bit further. So here's a peripheral protein. It's on the external portion of the cell. Here's another peripheral protein. This is inside the cell, but they don't expand across the membrane. Here are two integral proteins, and these actually extend all the way through the plasma membrane. So let's look at some of the functions of membrane proteins. There are quite a few different functions, and they're really important for the cell through communication or binding sites in our first example. A hormone binding site specifically, there's a protein receptor, and it has a binding site with a specific signal molecule. And what this does is it causes a change to the protein, usually a change in the shape, and it releases some sort of chemical signal within the cell. And so if we have a protein here, and there's a signal molecule that comes and attaches to that binding site, a hormone signal molecule, it's going to cause that cell to release some sort of chemical signal or to do something. And this can be very important, kind of going back to our emergent properties of cells and the development properties of cells. A chemical signal such as this is going to cause the cell to actually do something because of this chemical signal. An immobilized enzyme, it's a protein built into the membrane, and it's used as a catalyst to carry out a function. Sometimes that can be multiple enzymes working together at the same time. It could be one, it could be multiple, but it's a protein that's within the membrane, and it helps to carry out some specific function that the cell is responsible for completing or doing. Cell adhesion is a really important component of membrane proteins, and it can be used to connect to another cell's membrane protein. Within your reading, you should recall the discussion of gap junctions or tight junctions. Well, cell adhesion through membrane proteins allows these two different types of gap junctions or tight junctions, and so it allows cells to actually connect to one another. Cell-to-cell communication, as we previously discussed with glycoproteins, can serve as indicators for other cells. Can also be some short-term bonding. Cell adhesion and forming gap junctions or tight junctions is more of a long-term solution to bonding for cells, but cell-to-cell communication through glycoproteins can form some short-term bonding. We're going to discuss passive transport more in the next video, but membrane proteins allow for molecules to pass through the membrane. And this can occur through a protein channel, and it doesn't require energy. One of the primary things about passive transport that we're going to look at a little bit further in the next video is that it doesn't require energy. No ATP is required for this to occur. Hopefully you remember this from previous biology classes. Simple diffusion, facilitated diffusion we'll look at more in the next video. But membrane proteins are important in allowing molecules to pass through the membrane. And again, some examples, simple diffusion and facilitated diffusions are examples. Membrane proteins can also be used for active transport in that they can move substances across the membrane against the concentration gradient. by changing the shape through use of ATP. So again, we talked about ATP being that energy that cells use. Well, to pump something across the membrane requires ATP, and a great example of this is the sodium-potassium pump. And so we're moving something, in our example here, uh, sodium-potassium pump, we're moving something across the membrane, requires ATP. We're moving from an area of low concentration to high concentration rather than an area from high concentration to low concentration in passive transport. And so those are some of the different functions of membrane proteins and a quick overview of the plasma membrane. In our next video, we'll look more specifically at transport across the membrane, specifically looking at osmosis, diffusion through passive transport, and then active.